Oh no, I will detain it. Let me grasp it with my arms.
Is that a bomb?
Yes.
I better get some help
Saito,
with Chloe.
go tell Chloe
Okay.
that this is CTU's main bomb detainer. And we have an external threat.
Okay,
Tell her.
Chloe. There's a bomb. We gotta get contact with Jack.
Jack,
Oh, yeah.
there's a bomb. You gotta send your crew.
All right, we're on it.
Wait,
All right.
Chloe, I'm your biggest fan. Tell me what you do. I'll keep it in my memory for later.
All right, team. We need to detain the bomb. You ready, bomb squad? Follow me. There's bad guys in this building. Look, I think I see it.
I will open the bomb. The bomb is now open.
Alright. I'll go in and make sure that the whole thing gets destroyed. Alright, the bomb's completely destroyed now.
We must take it back to CTU and catch the man in charge of all of this. Meet back in 5 minutes. Chloe, we go. All right. I think we found the terrorist. Carl, I think you need to interrogate him to make sure that he doesn't repeat this vicious crime.
Okay. With my memory, I will go and interrogate one person over here. So, have you carried any bombs lately? Or,
Yes.
you have?
I have.
Guys, we found them. Alright, come with me, sir.
No. With your arms.
Okay, now wait a second. We've got to have Q&A, so you guys stand up here. Since the presentation was like totally lacking in any sort of content, we need you to explain everything because you don't have any labels. We didn't know what happened. And they're evaluating you based on how well you showed them that pathway. And they have the whole sequence of events with all the terminology and everything like that. So now you guys really have to go ahead and explain it thoroughly for us now. So who wants to...
So first we'll explain who we are.
Yeah, that'd be good.
Alright, so I was a virus, and I represented a virus, and I was carrying an antigen that was the bomb.
I was the MHC, binded with who was a macrophage. And
No, and you got to talk louder.
I was at MHC, I was binding with Will, who was a macrophage.
And I was the macrophage who swallowed the bomb and released the Saito. And I was
No, wait, where was the MHC?
That was Brandon.
I know it was Brandon, but in real life, where is the MHC? How do
It's inside the cell, then it brings it to the surface.
on which cell? You were a macrophage. What? You gotta explain. You gotta explain it to us. So the macrophage swallowed the virus.
No, I was inside of Will, who was
Yeah.
the macrophage.
And you're the MHC.
Yeah.
So inside of Will, who's the macrophage? Okay.
yeah, and I was the cytokine, which is responsible for carrying the message out
But where did you come from?
from Will to the macrophage,
Okay, so the macrophage released cytokines.
which alerted the helper cell.
And I was the helper T cell,
Mm-hmm.
which sent the message along to the killer cell, or killer T, which was
Wait, what message? How is a message sent from a helper T to a killer T?
through chemical signals.
Well, I received the message as the killer T, as I had a gun, killer T, and I told my helpers.
Well, the entertainment value was good. We liked it, but we have to learn stuff too. So we got to get the rest of
I
this.
was perforin. I'm secreted from the like killer T cells, and my job is like to go into the cell and place in like the pores of it and open them up.
So then I was the granzymes, so then I would go into the cell and make it either lysed by apoptosis or just
What were you again?
the granzymes
Okay.
that were released by the killer T cells.
And I was the memory T cell. Alice divided and formed me because she was a helper T cell, and she formed me on the memory T cell. So I can fight later infections.
Okay. So I think we need to go over this. Is anybody else confused? We got all the roles, but we need to know like the whole sequence of events again. So, okay, we got the macrophage. What is the macrophage eating? We don't understand that.
It's an antigen fragment.
It's eating antigen fragments?
Isn't it like presented to the macrophage?
What's it?
The antigen fragments.
Are presented to the macrophage by like the president, or you got to help us out here. Maybe you guys can ask some questions too to help clarify. So, Masha?
Well, I watched a wiki video, and the macrophage engulfs the antigen, and then it just breaks it up inside.
But what's the antigen? What's an antigen? Yeah.
It's a protein or a polysaccharide secreted by a virus.
And it's like the identification.
It's not necessarily secreted. It's part of the structure of the thing.
Yeah.
Whether it's a virus or bacteria or even our own cells have antigens. These are the way that we identify cells. It's stuff that's stuck in cell membranes, right? Or if it's a virus that doesn't have a cell membrane, it's just something that makes it up. So there's just antigens floating around. There's just parts of invaders floating around that the macrophage is going to eat. Is that what I'm hearing? Okay, no, that's not right. Okay. So the macrophage, that's our general cop that eats an invader like a bacteria cell. And then that breaks up the bacteria cell, and the little pieces are the antigens which bind to the MHCs and are put onto the cell membrane. So you didn't have it totally wrong at the beginning. You just had it somehow eating just antigens that are floating around. The macrophage's job is to eat whatever the invader is, and by the way it eats it by endocytosis, then the lysosomes inside the cell, those are enzymes, they break them apart, and then the antigens bind to the MHCs and go to the cell membrane. The macrophage becomes an antigen-presenting cell. Then after the macrophage becomes an antigen-presenting cell, what did we say happened next? So macrophage releases cytokines, the chemical messengers. And where do those go?
The helper T's.
To the helper T's. Okay. And so are the helper T's also binding to the macrophage at the same time? Yes. Because all the helper T's, and this is a really important point, all the T cells and the B cells in your immune system, they're not identical to one another. They're all slightly different because they have something called somatic rearrangement. When they divide, the genes get shuffled a little bit. So it's not true mitosis where you get clone cells. They're all slightly different. So what happens is a certain type of helper T binds with the antigen-presenting cell, and then when that binding happens, that's how you know you have the right helper T. Then the macrophage releases cytokines, and then that stimulates the helper T to do something now. So now that you know that you have the right general, the right helper T, what's going to happen to the helper T? Audience now. Yeah.
Is it going to divide rapidly and make more of itself?
Yeah. First it's going to be activated because these generals, they just kind of float around unless they're activated. And once they're activated, then they'll proliferate. And now you'll have a lot of these exact same type of general. Now you're cloning the general. And you've got lots and lots of these generals. And now they're moving out, and they've got the right types of receptors. And then they'll go ahead and bind with any antigens that are on any other surfaces of cells, whether it's on the surface of a macrophage, whether it's on the surface of, say, a normal cell, it'll bind. And then those helper Ts will start releasing cytokines, which will recruit killer Ts. Bind with the right killer T and make sure it has the right killer T and make the killer Ts proliferate. And then when you have the killer Ts, then they're binding with that specific pathogen and killing those specific cells. So it's all about specificity. It's really evolution in action, if you think about it. We have all these cells that are slightly different. There's variation in the actually genes of these cells, of these T cells. And it's about finding the right fit. And once you find the right fit, then there's cell signaling. And then there's proliferation. And you're making more of the generals and more of the killers to get that specific pathogen taken out. Yeah?
So, there are two types of T cells, right? There's like helper Ts and like killer Ts. So, is all a helper T do is like signal things that can kill the virus?
Right.
Okay.
So the helper T is the general, it's coordinating, and then the helper T will stimulate the proliferation of the killer Ts, and those are specific killers. They will only kill any sort of cell that's showing the antigens for that particular invader. And they don't get killed, they don't kill anything until they get activated by the helper Ts. Yeah?
If the antigen was just expressed from, like, with the class 1 MHC on the cell membrane, would the organism...
You mean presented to the outside?
Yeah. Then it was just left alone, no helper Ts came, would it just, like, eventually the virus overtake the cell?
Yeah, absolutely. So when the virus goes inside, some of the pieces are presented on the outside. But then, yeah, if that cell's not taken out, it could actually kill the cell and release more viral particles to go infect other cells. And that often happens. And there's sometimes viruses have special adaptations where they can cloak their antigens, and their antigens look very much like self-antigens in humans, and so they evade detection by the immune system. Or they find a way to not have their antigens presented on the cells in the first place. So they're effectively like hiding inside of cells where the immune system can't figure out what's in there. Okay, so what other questions do we have from the audience? Looking down the whole sequence of events. For you guys, just so you know, and you'll see it when you come back, the sequence was virus infects the cell, then the MHC binds to the antigen, is transported to the cell surface, then cytotoxic T recognizes that, then you have CD8 enhances the binding, the killer T is activated, helper T stimulates cytotoxic T with a cytokine called interleukin-2. The cytotoxic T releases perforin to go ahead and pop the cell, and the infected cell lyses. But you could also start it out the way you guys did with a macrophage. But you had the little hiccup where you didn't have the right sequence with the macrophage there. And then the other thing I noticed was the memory Ts. It's really important to know where those memories come from. The memory Ts come from a specific helper T that was involved in a response. After the response is over, those specific helper Ts, a few of them remain around as memory helper Ts. And then you've got memory cytotoxic Ts. So the idea is that invader comes again. You don't have to go through the whole process of trying to figure out the exact fit, because you already have these memory cells floating around with the exact fit, the receptor fitting those antigens. Any other questions from the group out here? Remember, this is it. You got a test on Thursday. You've got to know how these things start. You got to know how both these pathways are connected, and to be able to explain the specifics here in detail. Yeah?
So what was Alice's role again?
I was the helper T that sent another set of cytokines to the killer.
Right, and so that's right. Yeah?
Where would the CD8 be in that situation?
The CD8, it's a protein that's on helper Ts that helps it bind to the macrophage.
Yeah, and CD8 versus CD4 is not a part of the targets, so don't really worry too much about that. CD8 enhances binding of cytotoxic Ts, CD4 enhances binding of helper Ts. But that's not important. They're not on the targets. There's just some level of detail we put in the skit thing right here in the sequence of events. All right, well, thank you.